talk.
All righty. Hi, everybody. My name is Shaiva Patel, and as mentioned earlier, my faculty mentor is Dr. Tara Norgren, and today I will be talking about the bifunctionality of PRDX6 contributing to mediating oxidative stress in agricultural-related respiratory illness mechanisms via a pro-inflammatory response. So just because this title is so long, we're going to focus a bit about the introduction so everyone can get on board with what we are going to be discussing about. So the risk of respiratory illness is prevalent across all ages. Thus, it's important to conduct further studies regarding the immune response mechanisms that comprise the etiological origins of many of these diseases. Similar to any other processes, constant activation of the protective bodily mechanisms in response to external stimuli can be detrimental and can lead to diseases. Some common respiratory illnesses include chronic obstructive pulmonary disease, also known as COPD, acute lung injury, ALI, pulmonary fibrosis, lung cancer, pulmonary mycotoxicosis, also known as organic dust toxicity syndrome, chronic bronchitis, and asthma. In all of these examples, exposure and inhalation of organic dust are a common cause of disease. The overall pathway leading to disease includes the inhalation of dust particles that interact with resident lung structural immune cells, causing initiation of a deleterious inflammatory immune cascade that degenerates to tissue injury and disease when inappropriately controlled by proactive anti-inflammatory pro-resolution mechanisms. Some examples of these endogenous anti-inflammatory agents are antioxidant enzyme proteins and pro-resolving lipid mediators that are produced to regulate processes of inflammation resolution. As such, it is important to understand the pro-inflammatory and anti-inflammatory response in maintaining homeostasis in the setting of organic dust exposure. So the peroxyredoxin family, or the PRDX, as I will be referring to them, are antioxidant enzyme proteins that play crucial roles in lipid peroxidation, repair, lung phospholipid metabolism, and inflammatory signaling. They also participate in cellular signaling by taking on roles of activators and/or inhibitors in several pathways. As amplifiers of inflammatory signaling, they can potentially execute a dual role as a pro and an anti-inflammatory mediators. To regulate the onset of inflammatory cascades, they play key roles in two processes: peroxide signaling and reactive oxygen species, also further known as ROS signaling. So speaking of ROS, the ROS family is made up of reactive oxygen radicals and non-radical derivatives of oxygen that are included in the signaling cascade by maintaining balance among healthy intracellular redox processes. Oxidative stress, primarily defined as an imbalance of ROS and antioxidants, can be harmful to pulmonary tissue and cells. Excessive ROS can lead to tissue injury by oxidizing cellular proteins and liquid peroxidation of cell membranes. So the bifunctionality of PRDX6 and why it's really cool, and I wanted to explore a bit further about the enzyme. So this bifunctional enzyme has two distinct active sites that express phospholipase A2, also known as AIPLA2, and peroxidase activity. AIPLA2 is attributed to several roles in a myriad of cellular responses, in that phospholipid digestion and metabolism, signal transduction, and host defense. For the peroxidase activity, PRDX6 regulates oxidative stress. Although PRDX6 generates ROS for cellular signaling, it also regulates the population to protect against excessive cellular H2O2 levels, also known as peroxidase. Lipid mediators. So lipid mediators are derived from omega-6 and omega-3 polyunsaturated fatty acids and enhance innate and adaptive immune activities. Although each lipid has a unique function once freed from the cell plasma membrane, the roles are mainly in regulating cell growth and inflammation via receptor-mediated pathways. However, we will be focusing on macrophage mediators in resolving inflammation, or simply put, merisins, a particular family of compounds specially derived from docosahexaenoic acid, which is a tongue twister, so we will refer to it as DHA. And these metabolites also act as substrates for synthesizing more potent lipid mediators that act as second messengers. Now, the focus of the study is to evaluate the impacts of organic dust exposure on airway epithelial alterations in inflammation and redox functions. The hypothesis is that upregulation in PRDX6 expression via peroxide signaling is mitigating the oxidative stress caused by an increase in the H2O2 levels in response to HDE, which is hog barn dust extract. Apologies, but I have provided a diagram that will be listing the procedure that we use, which will cover the three main topics of human bronchial epithelium cell culture, the SWOT-MS collection, and statistical analysis. So under serum-free culture conditions, we studied the primary human bronchial epithelial cells, also known as HBEC, isolated from human patients. The cells were grown on 10 milliliters cell culture flasks with a pre-coated coating medium that contained 30 micrograms per milliliter purified collagen. For 30 minutes in a humidified carbon dioxide incubator at 37 degrees Celsius, the cells were incubated with 5 milliliters of coating medium. The cells were grown in LHC9 medium until the 80% confluency needed for each of the 4 separate exposure studies was achieved. Including the control medium, the treatment induced airway inflammation by exposing the cells with 5% hog barn dust extract, HDE, for 6 hours. The HDE dosage was chosen based on previous lab findings that indicated 5% would safely create an inflammation response without the risk of cell disease. So after rinsing with 1x PBS, the treated cells were scraped in Triton X lysing buffer with protease inhibitors and subsequently collected for SWOT-MS. Furthermore, we removed 600 microliters of protein in lysates from 2 wells to be quantified by nanodrop. These were sent out to the proteomics core laboratory at the University of Nebraska Medical Center. With the use of quantitative proteomics by SWOT-MS and bioinformatic analyses, the proteomics core laboratory provided identification and quantification for a set of 1,595 proteins. So out of the raw data, we narrowed down the peroxyredoxin family based on the following: biological processes, molecular function, and reactome pathways using StringDB, which is one of Elixir's core data resources. Data were analyzed via z-test and p-value to determine the regulation and statistical significance. Coupled with the standard deviation provided by the z-test, the p-value determines the significance of the data. So the results, as you can see on the screen, is just the z-test and the p-value, just because everything else wouldn't fit on the screen. But in order to specifically identify HBEC proteins, we only considered samples that were overlapping from both groups and determined a quantitative expression ratio that was based on total ion intensity normalization. The results provided the basis to test for the upregulation and downregulation trends in order to determine the difference in expression between the control and dust treatment. It also allowed for the acceptance or rejection of the null hypotheses, which states that there would be no detectable statistical significance between both groups. The results are shown in the figure, where HDE control indicates the comparison between the HDE treated versus control groups, and HDE Merizin 1 indicates the comparison between HDE treated versus Merizin and HDE treated groups. When comparing the results of the statistical analysis between HDE control data and HDE Merizin 1 data to the overall HDE exposure, trends indicate an inverse direction in terms of regulation. A negative z-test when the untreated control HBEC is compared to HDE treated cells indicates downregulation of PRDX6 expression in the HDE treated cells. However, in contrast, the positive z-test shows an upregulated pattern for HDE and Merizin 1 treated cells in comparison to HDE treated cells. Furthermore, using the p-value parameter that p less than 0.05 is significant, the results indicate that HDE control data and HDE Merizin data are statistically significant. So the discussion and conclusion, and why this is even important to discuss in the first place. So the study supports the hypothesis. However, it is important to note that HDE alone is not the only contributor in influencing PRDX6 expression. Data suggests that Merizin 1 expression could be increasing PRDX6 expression when compared to cells that were only treated with HDE. The increase of PRDX6 pro-inflammatory response suggests that the external stimulus is one of the factors to influence the protein to maintain homeostasis by upregulating peroxide signaling and ROS signaling via redox reactions. This is important since the overproduction of ROS leads to oxidative stress, which is, again, an immune system response that has a significant impact on lung diseases. One of the major sources to generate ROS is by activating NADPH oxidase type 2, also known as NOX2, which is an enzyme complex that contains multiple components and is bound to the membrane. This pathway is specific to microvascular endothelium, responds to an altered mechanical stress, and shows that an AIPLA2 dependent generation of ROS is associated with cell proliferation, cell growth, and vascular remodeling. By combining the present and past studies, we can further hypothesize that the bifunctional roles of PRDX6 have an impact on the overall immune system. The enzyme may act as an antioxidant that regulates oxidative stress by dust exposure and by protecting the membrane against lipid peroxidation via repair mechanisms. And future directions. So we hope to assess biological significance of PRDX6 epithelial cell upregulation under oxidative injury and other similar inflammatory conditions using ROS signaling and peroxide signaling as mediators of the response. Additionally, another future study should continue to specifically examine the NOX2 system in context of lung diseases with alterations in AIPLA2 activity of PRDX6. I would just like to give my deepest gratitude to my faculty mentor, Dr. Tara Nordgren, for patiently guiding me in putting this project together. I am appreciative of her constant support and effective feedback throughout this process. Without her mentorship and support, this project would not have been possible, truthfully. Thank you guys. Any questions?
Thank you.